Good afternoon. So my name is Joe Wu at Stanford University, and my co-moderator is Deanna Kroetz at UCSF. We're delighted to have you in this session on biologics and big data. And today we have four dynamic, outstanding speakers. The first speaker is Molly Jeffery at the Mayo Clinic, and she will speak on asthma biologics in the real world of clinical practice. The second speaker is Dr. Tony Wang at the FDA, and he will speak on understanding the use of animal models in evaluating SARS-CoV-2 vaccine efficacy. The third speaker is Dr. Richard Forshee, also at the FDA, and he will speak on risk modeling and the use of large databases for biologics. And then the fourth speaker is Dr. Russ Altman at Stanford, and he'll speak on quantifying the severity of adverse drug reactions using social media.
Hi, my name is Molly Jeffery. I'm the co-PI of the Yale Mayo CERSI and a researcher at Mayo Clinic. And I'm going to talk about some of our work on asthma biologics in the real world of clinical practice. This work was funded by a grant from the NHLBI, and the core team, who are all from Mayo Clinic, are listed here. They're an outstanding team. So claims data is a scrap heap. It's a big pile of refuse. By the time I see it, it has already served its purpose, which is to support the medical billing process, right? And ensure on the one end that the provider gets reimbursed for the services rendered, and on the other end, that the insurance company knows what the provider is entitled to be paid. At one point, each of the little data points that we work with was an actual human being who went to a doctor's office or to the hospital and received some care. But by the time we get it, that interaction has been emptied of essential content. It's been crumpled up. It's been stored away with hundreds of millions or billions of other shells of healthcare encounters, right? And our work is to recycle it. To grab those mangled shells and turn them into useful information that tells us something true that we didn't previously know. And further, our work is also to determine when claims data and other real-world data are the best and sometimes the only way to answer important research questions and clinical questions. So if we have big data, like claims data, we must also have little data, which is what I would call prospectively collected clinical and patient-reported data that requires researchers to talk to and potentially measure in various ways each individual study participant one by one. So if claims data are the crumpled, empty plastic bottles in the picture, then little data are this beautiful gem-infused crystal water bottle available from Neiman Marcus. They are beautiful, they are complete, they are very expensive, and they're representative of only a very small proportion of all the water out there to drink. For the past 5 years, I have been part of a team. We are led by Matt Rank, who's an allergist and immunologist at Mayo Clinic in Arizona, looking into asthma biologic use and focusing specifically on questions that can only be answered with big secondary data sets. All of the work I'm going to show you, it was completed using the Optum Labs data warehouse. The first asthma biologic treatment was introduced in 2003. Omalizumab was first labeled for use in people with allergic asthma not controlled by inhaled corticosteroid therapy. Biologics, as you all know, are substantially more expensive than inhaled steroids, and they are arguably also more onerous for people to use. And because most of them, including omalizumab, are administered in a doctor's office and generally every 2 to 4 weeks. Omalizumab was the only asthma biologic available from 2003 until 2015, but there are now a total of 5 available, and they differ a little bit in various ways. In our first study, we were really just looking at the prevalence and incidence of omalizumab use and comparing people with asthma who got the biologic to those who did not. Overall, as you can see in this this slide, which shows you the prevalence of only use, the quarterly use rate was quite low. It was about 2 people per 1,000 with asthma. And this is much lower than the proportion of all people with asthma who might benefit from use, which would be maybe 5 to 10% of people with asthma. That would be people whose asthma is not controlled and who have allergic asthma. When we looked to see which patients used omalizumab, we found some evidence of what we called suboptimal patient selection. So labeling required patients, as I said, to have poor control despite an adequate trial of inhaled corticosteroids. But when we used our pharmacy claims to actually calculate a measure of medication adherence to the inhaled steroids, we calculated the medication possession ratio for the 12 months before starting on OMA. We found that nearly half of the people starting omalizumab had very low adherence to their inhaled steroid, meaning they had filled less than half as much medication as would be expected to cover that 1-year time period. So this probably suggests that they were not using their medication as prescribed. But this is well known, right? We know that patients often have a difficult time using inhaled corticosteroids as often as they are intended to. The drugs can be expensive. They have to be taken every day, regardless of how people are feeling. Sometimes the regimen is complicated and people don't fully understand when they're supposed to use their rescue inhalers versus their regular controllers. So in work published this year, we revisited this adherence question and turned it around a bit. We were looking at factors that were associated with adherence to asthma biologics. So instead of just looking at the adherence to the steroids before starting the biologic, we're looking at what happens once you start the biologic. Because you might think that if people were not terribly regular in taking their medication that they can take at home, what's going to happen when they have to go to the doctor's office every 2 to 4 weeks? Will they be able to, adhere to that regimen? But we actually found that their prior adherence to the inhaled corticosteroids before starting the biologic was really only weakly associated with their adherence to their biologic treatment regimen. And the biologic treatment adherence was actually much better than their adherence to their inhaled corticosteroid therapy. So the average percent days covered, which is another claims computed measure of adherence, was 76% for biologics versus about 44% for the exact same people before starting the biologic when they were just using the inhaled steroids. So their adherence to their regimen went from 44% to 76%, despite the additional complexities of using a biologic. This team has also used claims data to better understand some additional questions. So how long do people continue on asthma biologics, which is their persistence in use, which is really important given the high cost of the treatment and the high burden in other ways for patients besides the cost, where patients have to visit the doctor so often to receive the treatment. And then we looked at what happens when people stop using asthma biologics. Do they have really poor outcomes? Do they start having exacerbations after they stop? And maybe most importantly, in a paper led by our really outstanding medical student, Regina Lamb, we looked at trial eligibility and outcomes when we compared people in our data set who would and would not be eligible for the trial for the asthma biologic that we observed them using in the data, to the extent that we can tell. We found that fewer than 1 in 10 people actually treated with biologics would have been eligible for the trial of that biologic. Just 1 in 10, less than 1 in 10. And in the cases where we could make comparisons using our data, when we looked at outcomes, people who wouldn't have been eligible for the trial of their drug had worse outcomes. They did not see the same benefits from those drugs as the people in our data who would have been eligible for the trial. So big data, little data. I mean, obviously we realize we need both, and I'm sure you all have a perspective on what's important about each, but here's my perspective. So 4 out of the 5 studies that I highlighted here, along with some additional work on asthma during the COVID pandemic, were completed on an R21 grant. So for those of you who don't apply for grants, that is a small budget 2-year grant, very short, small budget. None of the studies, so we got 4 studies plus a couple more out of this grant. We would have been able to complete 0 of them on this budget and this timeline if we'd had to collect primary data. And arguably, really, these were better studies for having been done using secondary data, because we weren't limited to a few academic medical centers or just to participants who had interest and capacity to participate in a study. You know, our measures of medication use aren't perfect, but the only perfect way to measure medication use is to go observe people using their meds. And you can't do that on a large scale with a representative group of participants. But maybe the greatest argument for the use of secondary data is we can get at outcomes and safety issues that simply are not possible with little data, as we see in our trial generalizability study. On the other hand, we can only see healthcare use. We can't ask patients if they missed less school or work when they used the biologic, or if they were unable to afford to fill their meds, or why they wanted to switch. And if you believe there's no causation without manipulation, as lots of people do, then little data is our only way to measure causal effects. So clearly we need both. And I would argue, actually, just to close out, that we are rapidly moving toward a new model that combines the two by linking prospectively collected primary data with retrospectively collected secondary data on a large scale. There are already tools, there are already research models in place that let us do this. And that, I would say, is where the future of clinical research is. Thank you very much.
Good afternoon. My name is Tony Wang. I am a research reviewer in the Division of Viral Products here at CBER FDA. I would like to spend the next 10 minutes covering the topic on the use of animal models in evaluating SARS-CoV-2 vaccines. First, I would like to declare that I have no conflict of interest and that the content of this presentation does not represent the official opinion of the FDA. When the pandemic hit us in early 2020, scientists around the globe rushed to develop tools to study SARS-CoV-2. Many groups, including our group here at the FDA, soon discovered that SARS-CoV-2 efficiently uses ACE2 of many species, from bats to human, to infect cells. The images shown in this slide were obtained by infecting cells expressing ACE2 of different animal species with a SARS-CoV-2 reporter virus. Upon infection, will turn cells green. And one can clearly see that ACE2 of nearly all species tested here, except mouse ACE2, permitted infection. And this observation made by us and also by many other groups has really laid the foundation for the development of multiple animal models. Today, 4 animal species are commonly used as models to study COVID-19 diseases, including mouse, Syrian hamster, ferret, and a non-human primate. Each model has its own advantages and shortcomings. For instance, infection of the human ACE2 transgenic mice by SARS-CoV-2 results in lethality, making it a very convenient model in testing efficacy of antivirus. However, the infected transgenic mice die of encephalopathy, which rarely happens in humans, and therefore there have been some doubts on how useful this model in studying human COVID-19 diseases. Then both ferret and non-human primates develop a very mild disease after infection, making them less ideal in understanding the COVID-19 disease. So to date, Syrian hamsters are perhaps the most used animal model to study SARS-CoV-2, particularly when it comes to study disease severity, pathogenesis, and transmission of the virus. Upon infection, Syrian hamsters may quickly lose body weight depending on the challenge dose, and develop viral pneumonia between day 4 and day 7. Hamsters also can be infected by close contact or airborne transmission, mimicking the transmission in human population. This unique feature allowed us to conduct a study last year to address one of the most important questions regarding SARS-CoV-2, which is whether prior infection will likely prevent a transmission. So illustrated in this figure, when a convalescent individual, the subject highlighted in green, becomes re-exposed to a virus carrier, highlighted in red here, there are two possible scenarios. One, the immunity that the subject in green has acquired from prior infection not only protects him or her from getting pneumonia, but also prevents transmission to another naive individual, because the subject has the immunity the subject has acquired really stopped the virus from replicating in the upper respiratory tract. Hence, this subject sheds no or very little virus. Under such a scenario, the spread of the virus through the subject is virtually stopped. Under the second scenario, the subject is protected from developing severe disease, but it continues to shed a virus because the acquired immunity in the nasal cavity or in the upper respiratory tract is not strong enough to stop the replication. Therefore, the subject continues to shed a virus despite maybe to a lower level to the community. So when a naive individual comes into contact, the transmission continues. Under such a scenario, the transmission of the virus in the community will never be stopped. We were able to reproduce both scenarios using Syrian hamsters, suggesting that even convalescent individuals may be infected in the upper respiratory tract. Worth mentioning is that as new variants continue to emerge, new hosts may be found. Using Omicron as an example, we have acquired new evidence that Omicron variant utilized mouse ACE2 much more efficiently than the ancestral strains. Green cells here are the ones infected by pseudovirus bearing Omicron spike protein. When mouse ACE2 is expressed, one can see massive infection. Interestingly, Omicron did lose the ability to utilize horseshoe bed ACE2 to infect cells, suggesting the change of its host range. Animal models serve an important role in evaluating vaccine safety and efficacy. When FDA issued the guidance document last year to vaccine developers, one of the biggest concerns was the potential risks for a vaccine to induce antibody-dependent enhanced infection or vaccine-associated enhanced respiratory diseases. We know this from our past experience in dealing with various vaccine products for RSV, SARS-CoV, or MERS-CoV. It turns out that a lower ratio of neutralizing antibody over binding antibody tends to induce antibody-dependent enhanced infection, whereas a Th2 type of immune response elicited by a vaccine tends to lead to vaccine-enhanced respiratory disease. Consequently, FDA recommended vaccine developers to evaluate these features in animal models first, such as a developer can really assess the ratio of IgG2A over IgG1, or the production of interferon gamma, or the ratio of neutralizing antibody to binding antibodies in the immunogenicity study in animal models. A vaccine developer can also evaluate the histopathological change associated with vaccination. They can directly assess the Th1 versus Th2 response after vaccination in animals. All of this will give FDA reviewers great confidence with regard to the safety of a potential vaccine. When it comes to vaccine efficacy, animal models also provide important tools in assessing both the immunogenicity of a vaccine as well as the protective efficacy of a vaccine in an animal challenge study. As a matter of fact, all three major COVID-19 vaccine developers submitted and eventually published animal studies of their vaccines. What remains challenging to vaccine evaluation is whether there is a true immune correlate of protection that can be used to rapidly assess the effectiveness of a vaccine. And I'm sure many of you are familiar with results like the one shown in the slide. It is quite often nowadays for us to hear in the news that a vaccine became less effective against a new variant because of the reduction in neutralizing ability. Yet, yes, neutralizing antibody levels are highly predictive of immune protection, as demonstrated in this elegant study by Miles Davenport. The predicted efficacy of a vaccine based on the neutralizing antibody titer it induces correlates with the observed vaccine in real world. However, as we understand the complexity of immune system, more animal studies in this area are urgently needed. Lastly, I'd like to throw at you two important notions which I felt quite often being overlooked when we discuss vaccine efficacy. Vaccine efficacy changes over time, over the emergence of new variants, such as Omicron, and over the change of status of an individual's immunity. So first, when do we know we need a new vaccine? Can we quantify this need in animal models? And secondly, many studies, including some of which were done in our own lab, suggest that the current vaccines tend to elicit an immunity that is much more protective against the infection of lower respiratory tract than infection of upper respiratory tract. Ideally, one would expect a vaccine that can induce a strong enough mucosal immunity in the upper respiratory tract such that after vaccination, the vaccinees would shed no virus or little virus upon reinfection. And this would indeed perhaps put the pandemic behind. The question is, can we determine efficacy in preventing transmission in animal model over time? Only time will tell. With that, I would like to end my presentation here. Thank you very much for your attention.
Hello, everyone. I'm Rich Forshee. I'm the Acting Deputy Director for the Office of Biostatistics and Epidemiology in FDA CBER. And today I'll be talking about risk modeling and the use of large databases for biologics. I want to start with a quote I like from John Allen Paulos. Uncertainty is the only certainty there is, and knowing how to live with insecurity is the only security. I use this quote here because it emphasizes that uncertainty is a fact of life that we have to deal with in making decisions. I'm going to be talking about how real-world evidence and quantitative benefit risk assessment can be used to help us better understand and quantify some of the uncertainty that we have when we're making important decisions. Now, the FDA has always had to integrate data from multiple sources in order to make our decisions. The poster that I'm showing here is actually on the FDA White Oak campus near our main cafeteria, and it was developed for Senate testimony back in 1964. The FDA commissioner at that time, George Larick, used this chart to illustrate the length to which, in deciding whether to approve or disapprove a given proposal, FDA reaches beyond its own staff to obtain data and advice. Now, of course, one of the major issues that we're dealing with at the FDA and in particular at CBER right now is the safety and effectiveness of the COVID-19 vaccines that are used in the United States currently. And real-world evidence has already played a big role in understanding the safety and effectiveness of these vaccines. We've used many systems, including the Biologics Effectiveness and Safety System, or the BEST system, for gathering data on these vaccines. Real-world evidence has been promoted through the 21st Century Cures Act, which was passed in 2016. And this act places additional focus on the use of these types of data to support regulatory decision-making, including approval of new indications for approved drugs. I want to take just a moment to talk about my team's approach to real-world evidence. And this really starts with asking the right question. You have to make sure that the question that you're looking at is something that is relevant to the public health mission of the FDA and that it's a feasible question that you can get an answer to in a timely manner. Once you have the question right, it's important to then identify fit for purpose data that is appropriate for addressing that question. And you also need to minimize bias by design. This is done in the protocol phase, where you carefully consider the potential sources of bias and make choices in order to minimize this at this early stage of the study. As you begin to conduct the analysis, you also need to plan for many sensitivity analysis, as well as to identify and quantify the remaining bias once the study is complete. Now, this process is not a linear process that ends when your study is complete, but rather oftentimes additional questions will be identified as you conduct the sensitivity analyses and identify and quantify remaining bias, leading you to return to the design stage for future work. Benefit risk assessment has also been a focus at the FDA over the last several years. As part of the Prescription Drug User Fee Act 6, the FDA has committed to furthering the agency's implementation of structured benefit risk assessment into the Human Drug Review Program. And we have many efforts underway, including working on guidances with the FDA and working with international groups in order to advance benefit risk assessment. What I hope we'll be able to discuss in more detail during the panel discussion of this session is the intersection of real-world evidence and benefit risk assessment. Real-world evidence will provide many of the key inputs for benefit risk assessment, and the tools of benefit risk assessment can help us understand the strengths and limitations of the real-world evidence and integrate it into a decision-making process. There are many important uses for benefit risk assessment tools. One of the most important is establishing an agreement on the key benefits and risks. Tools also exist to summarize key evidence, to integrate data from multiple sources. We have tools to help us evaluate the impact of uncertainty on the decision. We have tools to help us estimate benefits and risks with uncertainty. We can help understand the relative importance of benefits and risks through processes such as applying weights to benefits and risks. And we can use the benefit risk assessment to compare estimates for the outcomes under different regulatory options. Benefit risk assessment tools can also help to improve communication with other stakeholders. The very process of creating a model forces everyone to be clear about the benefit, risks, and evidence under consideration, along with the assumptions that go into the model. Visual and graphic tools are also available to enable scientists and the public to examine a complex decision. And these visual representations can communicate complex relationships of benefit and risk that can be critical to FDA decision-making. In conclusion, there's a variety of benefit risk assessment tools that are available to help people carefully examine a difficult decision. The modeling effort itself can and should be tailored to the specific decision. Benefit risk assessment models can help to integrate many sources of evidence and quantify remaining uncertainty. And the very process of developing a model is extremely valuable. So thank you for your time. And I'll end with another photo from the FDA White Oak campus. There are several benches throughout the campus that have statements from court cases. This one is FDA insists reality matters. Thank you very much.
Hi, this is Russ Altman from Stanford, and I'm happy to share my screen and tell you about some of our recent work. This is quantifying the severity of adverse drug reactions using social media. This was part of Adam Levertu's PhD and was also worked on by Taimur Hamamsi, who's now at Columbia. So we know about pharmacovigilance systems for adverse events. The FAIRS system at the FDA, the Sentinel Initiative, all give us information about adverse events associated with drugs and a sense of their severity. We're interested in looking at social media for understanding the severity of adverse events. Whereas existing sources focus primarily on data generated with the healthcare system, social media comes from the patients really directly, but there are challenges with it. The relevant information has to be identified and extracted from text. People use slang, misspellings. They are not professionals. Only a small fraction of the data in social media is relevant to pharmacovigilance. The description of adverse reactions can be unclear, and other problems as well. We're going to be looking at Reddit. It's a very large community-moderated social media platform, which is organized into topics. Some of the key topics are health, drugs, and other health-related topics that are on Reddit. In a Reddit discussion, somebody posts something. This is the drugs Reddit thread. Somebody here has posted something about COVID vaccine and cocaine. That's a submission. And then there are comments that follow it up. The comments are associated with usernames that are handles and that are not very identifying of the people, and they're certainly public. The thing about Reddit, however, is that the data exists since 2005. There's been 651 million submissions and 5.6 billion comments. These are numbers that force us to pay attention to it as a potential source of pharmacovigilance information. Now, in work that I'm not going to discuss here, we published a paper about a RedMed lexicon that is able to take everyday language that's used in Reddit and map that to formal medical terms. So in that paper, we showed that for both drugs and for adverse events, we could identify slang, misspellings, et cetera, and map them to the normal MEDRA terms that are often used for coding adverse events. And RedMed was able to do this for about 3,000 drugs and a large number of adverse events as well. And the reference is at the bottom. Now, I don't need to tell this group that adverse drug reactions are costly. 100,000 people per year in the United States, and they can cost up to $500 billion of annual spending in response to the complications that come from these adverse drug reactions. And as we also all know, ADRs are currently characterized on a per drug basis into the most severe, which are boxed warnings. And you see a boxed warning on the right, lower right there that says warning, life-threatening hematological adverse reactions. There are also warnings and precautions. And then the least severe is adverse reactions. So these are categories. This categorical system is limiting. The boundaries between the categories are not always clear. It's difficult to assess the importance of a new ADR and to know what the right category should be. It's difficult to compare drugs overall across all their uses and diseases. And so the use of these categorical systems is an impediment to some types of data science that might be useful. So we wanted to know if we could derive a quantitative severity score for ADRs. Now, in previous work, my group did this by paying crowd workers, that is workers on the internet, to make pairwise comparisons of ADRs. What's worse, a headache or cancer? What's worse, a rash or heart disease? And using these pairwise comparisons, we published an algorithm for ranking all the ADRs. And we did this in triplicate with different crowd workers with a very good correlation of about 0.71 across the three groups of workers. And we showed in that paper that the ADR severity correlated with the rate of death from those ADRs. However, this was imperfect. We only used about 3,000 of the 15,000 ADRs in FAIRS. It was time and labor intensive. The cost did not scale. And of course, the crowd workers were not medical professionals. So we are presenting a new way to do the severity of adverse events based on networks. So in the lower left, you see these black dots. Those are the ADRs that are connected to one another based on the similarity of the ADR, based on an analysis of the text that describes the ADR. So this is a network of similarity in ADRs. In that second picture at the bottom, you can see we've labeled a few of the ADRs in bright red that means they're very severe. That was done by hand. Suicide, death from heart attack, those are red. Then there's some blues, which are relatively benign, mild rash, mild itch. Those are in blue. We used an algorithm that I won't go into the details that then connected everything in the network based on whether it was closer to the red guys or closer to the blue guys to get an estimate of the side effect severity. This gave us our SAEDR, or a side effect severity of adverse events derived from Reddit score. And you can see the distribution of scores going from 0 to 1. Not surprisingly, most of them in the middle, but we do have ones that are very severe and ones that are less severe. We showed a strong correlation with our previous crowd worker estimates. At the lower left, you can see yawning, cold sweat, or dry tongue. And at the upper right, you can see neuroblastoma, cardiac death, and metastatic lung cancer. So very severe is on the upper right, less severe in the lower left. Our SATER scores show differences across disease groups. At the very top, you see cancer, cardiac disorders, and hepatobiliary disorders over all drugs used in those categories. And at the bottom, you see skin and subcutaneous tissue, general disorders of administration, or product issues, just less severe. We also can dig down. If you explode the cardiac disorders into cardiac neoplasms, coronary artery disease, heart failure, we can start to separate out the relative severity of these different adverse events. So how can we use this measure? Well, on the first two, we compared box warnings with no box warnings, and not surprisingly, the average SAEDR score for boxed warnings, the black violin plot, is on average much higher than things that don't have a boxed warning. We can compare the severity of side effects that are reported in males and females. We can look at clinical trials on the far right, post-market, and then two databases that we created in other work, and look at the average severity in those databases. There's a lot more we can do, but time limits my ability to describe this. One thing, however, is we created the DRIP score, the drug risk profile, that's an integrated estimate of the overall risk of a drug without considering the severity of the disease. So it's simply the frequency of the ADRs associated with that drug times the severity of those frequencies. And so, not surprisingly, anti-neoplastic and anti-epileptic agents have very high DRIP scores, again, without reference to what they're treating. So you, of course, would have to consider that in folding in DRIP scores. And then drugs used in diabetes are actually quite safe for the most part, on average. And of course, there are exceptions that we all know about. So these DRIP scores are a useful summary and move us towards a quantitative estimate of the severity of adverse events and a quantitative estimate of the risk of individual drugs. So these are all available. The SATER scores are available at this bit.ly URL for 12,000 ADRs. DRIP scores for almost 1,000 drugs are available at this URL. And really, my message is we want to enable a shift from categorical analysis of ADRs to quantitative analysis. And that would be great for all of us. Thanks very much.
Thank you for those great presentations. We'd now like to open this up for questions from all the attendees with our panelists here. So we'll start, so please add those through the question and answer function in Zoom. So we will start with Molly. And there's a question here about diversity and sex and how that was considered in whether the biologics helped or not for the patients that you were discussing.
It's a great question, and something that I think, would like to do in future work. In this particular set of studies that I presented, some of them we actually used matched cohorts, and so we weren't able to do that. And in other ones, it just wasn't the focus of our analysis, and so we didn't do it. You could potentially do it, though, with the data that we collected, and it would definitely be of interest. I will say that in terms of the one, I don't know if this is the one that you're referring to, but the one looking at whether people were eligible for studies or not. The primary reason that people were not eligible for studies was they weren't taking the right medications in advance, or they didn't have the high enough sort of percent days covered, or they didn't have enough exacerbations. And so it wasn't necessarily like personal characteristics. It was more sort of those aspects, those clinical aspects.
Thank you. Our second question is for Tony. Can you comment on Pfizer and Moderna vaccine preclinical dose escalation and virus challenge in animal models, and why there were only 2 doses that were tested for both of these, which is quite different than what we're used to in drug escalation studies?
Well, first of all, since I'm pretty sure that both companies performed animal studies, or particularly immunogenicity studies, using at least the 3 doses. As far as I recall, with Moderna, I think they did that in some immunogenicity study in mice using 0.01, 0.1, and 1 microgram. In their challenge study, you are correct. I believe both companies use only 2. But I had one slide during the presentation. The review process for both the mRNA products were a little bit unusual in the sense that we were in the pandemic. So the entire process kind of got, squeezed together. We pretty much allowed the developer to conduct the animal study, and as soon as some of the results are in, and they're allowed to go ahead for the phase 1, in which both companies, I believe, used at least the 3 doses. For Moderna, I think that was 25, 100, and 250. Then for Pfizer, I believe they also used the 3 doses in phase 1 to ensure safety. So just to make a long story short, first of all, I think these, this is a somewhat unusual situation. On the other hand, both companies did immunogenicity study in animals using at least the 3 doses.
So actually, I have a question for Richard. Richard, I enjoy your talk quite a bit on the real world evidence. And I wonder if you could give us a perspective on how do you evaluate how solid this data set is. And the main thing I'm pointing out to is, for example, as you may recall, early in the pandemic around 2020, there were 2 papers, one in Lancet and then the other one in New England Journal of Medicine. The one in Lancet was on anti-malarial drug, and then the one in New England Journal was on ACE inhibitors. Both papers were withdrawn, were retracted because they found the company CERSI essentially kind of made up the data. And so I just wonder, how do you adjudicate what's considered good data, what's considered bad data?
Thanks, Joe. I'll touch on this. A complete answer would take more than the time that we have left for the whole discussion. I will say that the FDA has been working on developing guidelines for how we're going to evaluate any evidence that is submitted, and particularly evidence on efficacy. We've used real world evidence to get information on safety aspects of drugs for decades, but now we're focusing more effort looking on using real world evidence for studies of effectiveness. And one of the key things that we've already identified is that we want to be sure that everything in a study for effectiveness that might have a regulatory purpose down the road needs to be pre-specified. And it's best if people reach out to the FDA early if they're planning a study like this that they hope will be usable for regulatory decision-making later, so that we're sure that people haven't simply sorted through the data and chosen the results that were most favorable for them. We want to make sure everything was done per protocol. And as I mentioned in my slide, it's also critically important that we find fit for purpose data. And this has to be evaluated on a question by question basis. You can't simply say that one data source, say Medicare, is universally fit for purpose. You have to evaluate this for each question that is being evaluated. So I think to summarize, the FDA is going to be providing more information on this. We are developing guidances on real world evidence. And one of the key elements is pre-specification via protocols, and then determining that the data itself is fit for purpose. And this includes being able to validate that data to make sure that it really exists and that the data that's being used reflect the data that were originally collected. I think I'll go ahead and stop there. I hope that was helpful. Thanks.
Got it, Richard. That was very helpful. So I have a question for Russ. Russ, I enjoy your talk on the social media, and being a Twitter user myself, I just wonder if you can comment on this amplification effect, meaning that if somebody who has 1 million followers and tweet that I took beta blocker and I have some issues, versus somebody who only has 50 followers and tweet the same thing and say I took beta blocker and I have some issues, and wouldn't it be disproportionately weighted heavily on somebody who has 1 million followers, because somebody will then be retweeting that person's message, whereas the second case with 50 followers, it's unlikely that that person's message will get retweeted.
Yeah, that's a great question, and we've looked at that a little bit. The first thing is that these social media outlets are different, and Twitter, I think, does have more amplification than Reddit. Reddit is a little bit more of a discussion, but in our very preliminary look, one of the things we noticed is that discussion of the topic goes up, but people don't all just fall in line. As you like, so somebody says, "Oh, drug X caused me, to have a seizure." People are the Redditors, at least, are naturally suspicious, and so although the discussion does go up, it's not like everybody is agreeing with that. So there's a certain dampening effect from the discussion. This requires a lot more work for obvious reasons. If we're going to take this as a serious data resource, we have to understand these biases and the dynamics of these kinds of things. The other thing that my student found is that Reddit is so big that it is hard for an influencer in Reddit. I don't think it's the same in Twitter. In Reddit, it is hard for one person to move the conversation as much as it is in Twitter. So this gives us a little bit of optimism, but this is actually, I think, the key research program going forward, because now that we've shown that you can do things with this data, the provenance and validity. One thing that I didn't discuss is we were able to show in following opioid epidemic comments that Reddit predicts trends in opioids that are later detected by the CDC. And Reddit, you can detect it in Reddit based on a historical analysis a couple of months before the CDC recognizes it. So there is great promise, but as you imply, these sources of bias need to be evaluated.
Thank you, Russ.
Hey, moving back to Molly, I think a question that's relevant for the CERSI crowd here is when you talk about the very low rate of success with these biologics in the real world, and compared to the trial populations that most of these people would not have qualified or been eligible for these trials. What is that, how does that inform us about trial design to begin with? Are we really understanding what are the populations that need this drug, and are those the ones that are being included in these trials?
Right. So it is a really important question. So as I kind of referred to, one of the main reasons that people weren't eligible for the trial, but were actually receiving the drug, was that they had too few asthma exacerbations. So that could suggest that their disease was less severe than what was thought of. And I think, I think part of the question is, so asthma biologics are very expensive. They're a lot more expensive than the drugs that people take at home. And so if people do have a difficult time for whatever reason, taking their home meds or keeping up with a more difficult regimen like that, but they are able to do it with biologics, then we can consider that to be successful, taking aside the question of economics. If what's happening is they're actually, more likely to be adherent, which we did see, and if it then reduces the types of issues that are causing them to want to switch to a different med, right? So we can't see in our data whether they go to school more often, whether they are able to sleep better, things like that. So it's a difficult question. I think what it really gets to is that you will always, once you put a drug out on the market, have people taking it who aren't in the exact population that were intended and that were studied. And so, are you going to expand trials to the extent that all comers are eligible? No, you're certainly not. So it's just a different question, I think. But it's an incredibly important question to understand, and so that physicians can understand, if I give this drug to a patient who isn't like the people in the trials, what's going to happen?
Thank you.
So Tony, I have a question on your slide with regard to the different animal models. That slide was actually very helpful for me, that summarizes these animal models. So I wonder, within the animal models, is it possible to predict what the next emerging variant or mutation of the virus will be?
It's a great question. I guess the answer I would say is probably yes or no. In fact, even without using animal models, just based on pure structure analysis, we can get a pretty good idea whether a new variant bearing certain mutations could be more or less infectious. Having said that, Omicron really sort of humbled me in the way that the mutations found in the RBD, in fact, appear to be detrimental to the binding to human ACE2, yet the virus appears to be more contagious. On the other hand, there are 3 mutations also in Omicron's spike protein. Based on the structure analysis and the past knowledge, we would think it'll increase the so-called fusogenic ability of the virus, which means to make the virus to fuse, to infect better. But then the experiment data, in fact, contradict the prediction. So ultimately, yes, when we have a new variant, we would like to put in a certain animal model. Right now, I can say that the Syrian hamster probably is the best or the most accurate model close to human for many reasons. But really to predict at a population level, I think that's a very challenging issue. I'm afraid I don't really have an answer for that. Because this virus basically just changed a lot of people's perspective. It does amazing things.
Thank you.
I think Richard, you had a comment that we didn't quite catch. Sorry.
Yeah, thanks so much. No, I just wanted to pick up on something that Molly has mentioned a couple of times now about the link between what she's calling little data and big data, or the randomized clinical trials and the kinds of claims or EHR data that we might want to use. I just want to say that I completely agree with Molly that these really need to be viewed as complementary ways of understanding these questions. Clinical trials are really elegant at answering some very specific questions in a way that we couldn't get at otherwise. But when we start working to generalize to other populations, when we start trying to get evidence about rare adverse event we really need to use some of the real world evidence that we have to build on what we've learned. But it's not like one is better or worse than the other. Each have their strengths and limitations. And if we link them together, we can get a more complete understanding. Thank you.
Thank you for that comment. Russ, I see you answering away here, but I think it would be great if you could comment on the questions that are coming in about how this could be applied. Is it best used post-marketing? Is it best used during development? Do you have any thoughts on that?
Yes, thanks very much. So post-marketing for me is a no-brainer because Reddit data is available. Of course, it's not the whole story, but it's a pretty good and interesting indication of the patient experience of these drugs. And so we would expect to and plan to continue monitoring. Every now and then we might actually discover an undiscovered adverse event that's coming up enough in the Reddit comments, and that seems to be real, that we could refer it for follow-up. It's an interesting question, and at least two people asked about what about in drug development. So first of all, if drug companies have like forums where the people involved in trials can just put in their experiences, and I don't know if they do, and they might not, because it could be a huge headache. But with Reddit type conversations among drug trial participants, and that's tricky. I mean, just saying it makes me think about all these issues. But it would be possible to try to understand the patient experience of these drugs in their own language. And then we could start to look at the severity scores that we've defined, which are quite empirical, and we could recalibrate and say, okay, this drug is looking like it's having more side effects than we expected. Or you could do it comparatively and say, this drug looks like it's having more side effects than this other drug had at a similar phase of development. And we could start building up a corpus of how do drugs look early. I believe that you probably get indications of the severity profile of a drug pretty early. As a clinician, I remember when the SSRIs came out, it was obvious to everybody that the profile of side effects was going to be more attractive than tricyclic antidepressants. And that didn't take a long time. And I learned that first from patients using the drugs than any studies that were published. So I think it's a component, but let's be realistic. This would potentially be a component of the data profile that could be generated. I think the benefits of it would be the patient experience would be driving these scores, and that would complement the biomedical and healthcare provider and more objective data collected during the trials, and even in real world evidence, as Molly and others have been talking about. This is a third higher variance, but providing a different projection of the experience for the drugs.
I think we are running out of time here, actually. I think we should end it here so people can log back in to the main session. Thank you all very much for these outstanding talks. I think there were a lot of very interesting questions that were raised by your talks. There was a lot of interest from the audience. We'll try to answer the other ones maybe before we log out here directly that we didn't have time to hear. And thank you again. So please, when you log out of this Zoom session, you need to go back to the main session, the same link that we used this morning by 1:30. So thank you everyone for participating.